New research from the Sanger Institute in Cambridge suggests that the genome of an individual cancer not only tells the story of how it developed, but also of how it could have been prevented. Mike Stratton, director of the Institute, told me more.
All cancers are believed to be due to mutations, to somatic mutations, mutations acquired over the course of a lifetime in cells of the body. But mutations don't just happen. Something has to cause mutations. And in principle, every single mutation we should be able to ascribe a cause of some sort to. So evidence and studies over the last 20, 30 years have already given us an indication that the mutations that we find within cancers reflect the processes, the exposures that caused cancer in the first place. And the best example of those are really in skin cancers and lung cancers, the effects of ultraviolet light and tobacco carcinogens, respectively. So if one looked, as we could do through the little keyhole of the p53 gene 20, 30 years ago at skin cancer, what we found was a particular predominance of C to T mutations, so conversions of cytosine to thymine, that we found in skin cancer, and this was compatible with the fact that ultraviolet light was also known to induce C to T mutations. Conversely, when people looked in lung cancer, they found a much larger number of C to A, cytosine to adenine mutations, and that's what we knew already tobacco carcinogens would produce. So it appeared that the genomes of cancers bore the mark, the imprint, what we now call the signature of the processes that caused them. So the opportunity to us now, compared to then, when all that we had was the p53 gene, now we can sequence hundreds, thousands of cancer genomes, and indeed at the moment there are about 30,000 cancer genomes sequenced. Now we have an opportunity to use that tidal wave of data to explore and define all the mutational signatures from all the mutational processes that are contributing to the mutations in cancer.
So how do you pick out a pattern that makes sense from such a mass of data?
Well, the data is complicated because for any individual cancer, the mutations that you find have been generated across the whole of a lifetime, and they can have been generated in one period, throughout the whole of life, or intermittently. Moreover, in a single cancer, as in indeed a single normal cell, multiple different mutational processes may have contributed to the mutations that you find at the point that you remove the cell or the cancer. So in order to analyze that sort of complexity of data, we have to make certain assumptions, have an underlying model, if you like. And that model is that there are multiple different mutational processes operative. Each mutational process results in its own signature, types of mutations. And then what we do is we compare thousands of cancer genomes under the assumption that the contribution of these processes and therefore signatures differs between each one of those individual cancer genomes. And then it becomes a standard but actually quite difficult mathematical problem. It's a problem like being in a room with multiple conversations going on, and what you actually want to do is to hear each conversation separately. You've got to extract them all out from each other's, because those multiple conversations are contributing to the hubbub, the noise in the room, and from that noise you now want to pull out all the individual conversations that have been contributing to that noise. That's a standard problem that the mathematicians have been going at for a while. For this particular purpose, there is a sort of algorithm, a mathematical approach called non-negative matrix factorization, which has features that lends itself to doing this, and that's what we used when we take thousands of cancer genomes and essentially extract out the conversations, and those conversations are those mutational signatures, the component parts of the mutational catalogues that are present.
And you got it down to a manageable number?
Yes. Currently there are about 40 different mutational signatures that we have found. it's not going to be an infinite number. Clearly, as time goes on, we're going to find more signatures that are attributable, for example, to local exposures, so small numbers of people to exposure. And we are going to find signatures that are associated with cancer treatments, because cancer treatments often are DNA damaging, and we already know that they leave their own imprint, their own signature on genomes, and there'll be quite a lot of those. But if we're talking about the mutational signatures that are present across the bulk of human cancers, these are 30 to 40 of these patterns, of these signatures, and we don't expect the number to increase dramatically from that.
What's the work told you so far in lung cancer?
Well, in lung cancer we have an established dogma, and it's been a pretty good dogma actually, which is that tobacco contains chemicals, tobacco smoke contains chemicals, and those chemicals damage DNA, and because of that DNA damage, cells that are exposed to tobacco and tobacco chemicals acquire mutations. So there is a mutational signature that has emerged very clearly as being associated with tobacco smoking. In our parlance, that's mutational signature 4. And it has a very distinctive pattern. And we know that it is associated with, or it is due to tobacco actually, I think we can say, because we find it only in cancers that are due to tobacco smoking, and indeed when we take some of those chemicals that we know are present in tobacco smoke and contribute to carcinogenesis, and we expose cells in vitro to those chemicals, then we find essentially the same signature. So that signature 4 clearly is associated with tobacco smoking. And indeed, when we look in lung adenocarcinoma and lung squamous carcinoma and laryngeal cancers, we can clearly see the presence of this mutational signature 4, and we can see more of it in previous smokers than in never smokers who have those cancer types. So all of that fits. The trouble is then that when you go to other cancer types, particularly those cancer types in which the normal cells from which they derive are not directly exposed to tobacco smoke, cancers such as bladder cancer, a long way away from inhalation of cigarette smoke, there's a 4-fold risk of developing bladder cancer if one is a smoker compared to being a non-smoker. But in bladder cancers, we do not find that signature 4. We have tried very hard to find it. We've tried to, in the mathematical analyses, tried to force it in, but there doesn't seem to be any place for it. And our best guess at the moment is that this signature 4, clearly associated with tobacco smoking, clearly due to tobacco smoke, is not found in bladder cancers, and it's not found in bladder cancers from smokers. So there needs to be something else that tobacco smoke is doing other than this signature 4 that is causing the bladder cancer and many other types of cancer. So we did a broad kind of mutational signature analysis of the 15 different types of cancer that are associated with tobacco smoking. And what we found is that there is one signature, a signature 5, which is increased in smokers compared to non-smokers for about 10 of those cancer types. Now this signature 5 is a rather mysterious beast. As a signature, it's not terribly distinctive. It's a rather sort of vague signature, sort of featureless signature. It's found in normal cells. It's found in probably all normal cells. It's an endogenously generated mutational signature that is part of just being human. But if one smokes, then it seems to be switched on more than it would be otherwise. And that generates an additional mutation load. And it is at least plausible that that additional mutation load in cells from the lung and the larynx and the pharynx and the esophagus going down into the bladder and the kidney, we see that increase in signature 5 in smokers compared to non-smokers in all cancers of all those types, that may be contributing, that may be responsible for the risk of cancer, of those types of cancer that tobacco smoking is causing.
So for some signatures you know the cause and for others you don't?
Yeah. So about a third to a half of the signatures at the moment, we can't even speculate on their causes. That's a smaller number than it was 2 to 3 years ago, when it was more like 2/3 of them. So gradually we're whittling away. Although, to be honest with you, quite a lot of these are speculations. But for us, it's better to have at least a speculation, a hypothesis that can be tested, than to have nothing at all that we can say. But yes, for still about a third of them, there is nothing at all that we can say. There are some telltale features about some of these mutational signatures that makes one suspect what they might be. And some of them do look like the sort of signature that you get from an intense exogenous DNA damaging exposure, such as this tobacco smoke chemicals, such as the chemical that is present in maize that's infected by fungus in Africa, so-called aflatoxin, which causes DNA damage, and some others. These are very, very highly mutagenic chemicals that cause particular patterns of mutation with particular features. And we have a few of those which are unattributed at the moment, and therefore we think that they must also be, or quite likely to be, due to some exposure of that nature. And obviously that's an important thing to explore, because if one could work out what the highly intense exposure is, then it should be preventable. And there are a number of growing examples now where the mutational signature analysis is beginning to lead us to these preventive strategies. After all, what we're after here now is the causes of cancer, and once we learn them, we want to prevent them. And one interesting story in this regard is with regard to this a chemical called aristolochic acid. This is an extract of a plant, plants of the Aristolochia genus. It's sometimes taken as a dietary supplement. It's sometimes found as a contaminant of foodstuffs. It causes a lot of mutations. It's highly mutagenic, this stuff, despite the fact that people ingest it voluntarily. And it causes an increasing range of cancers, more than was initially suspected. But in some recent studies that were done, looking at kidney cancer, to the surprise of the investigators, when they looked at the kidney cancers from Romania, they suddenly found all the kidney cancers from Romania had this signature in large numbers. This was completely unexpected. Nobody had any idea that people in Romania were being exposed to this aristolochic acid. The fact that this has emerged will lead on to much larger studies to investigate the extent of this exposure, but it immediately brings to the fore some form of public health action to reduce the exposure, to find out where the aristolochic acid is coming from. Is it a contaminant of foodstuffs? And then to remove it in order to remove the mutational exposure and hopefully to reduce cancer at the same time.
So what's next in research?
So the mutational signatures approach has, I think, a number of quite substantial aims. The first is that we should aim, now that we have thousands, in fact tens of thousands of cancer genomes, to get a comprehensive overview of the mutational processes and their signatures that are contributing across all different types of cancers. That's a first sort of milestone. Then we need to use this approach to explore some of the residual mysteries of cancer causation. There are a number of cancer types which show very, very dramatic geographical variation, 100-fold differences in incidence between different parts of the world. Despite these very dramatic differences, epidemiologists have been unable to work out what it is in one place that causes the high incidence and in the other place is not present, that is resulting in the low incidence. And this is despite many years of study. So one set of experiments to do now is to collect cancers from high incidence areas, collect cancers from low incidence areas, to collect epidemiological information associated with them, and then to sequence their genomes, and to compare the genomes of cancers from high incidence to low incidence areas, and to see if there are different mutational signatures in there. What may also emerge from that is greater understanding of some of the known risk factors for cancer in which we really don't fully understand the mechanism. For example, it's known that obesity is associated with an elevated cancer risk, but we don't really know how that is working. So in a similar way, by collecting cancers from individuals who are obese and individuals who are not obese, sequencing their genomes, we will see whether obesity and all the metabolic effects that are associated with it is contributing to cancer causation through a mutational process. It's unlikely to have its own very distinctive mutational processes, but it could be ramping up. It could be amplifying endogenous mutational processes which are present in all of us and just increasing the mutational burdens of some of those. And that could be responsible for the excess cancer risk associated with them. So there's these big alliance of mutational signature analysis with epidemiology to look at some of the grand problems of cancer epidemiology which are still left to resolve.
Thank you very much.